Welcome everyone to this Share Your AXA Research event. Today you're going to listen to postdoctoral fellows that will have 3 minutes to share their research to you, and trust me, it's not an easy task. The postdoctoral fellows you will listen to will have been selected following a call for projects on harmful substances launched last year by the AXA Research Fund. This AXA Research Fund Fellowship aims to build a solid multidisciplinary understanding of the effects of contaminants on human health and the environment and find ways to mitigate these negative impacts. The ultimate goal is to inform industrial and risk management strategy to enable public policy design in this area for a safer human and environmental health. The selected fellows come from 4 continents and have started or are about to start their AXA-supported research projects in leading universities from all around the world: UK, France, Australia, Finland, and Luxembourg. 2 of them couldn't make it, so you will have pre-recorded videos. The mission of the AXA Research Fund is not only to fund excellent, innovative risk science, but also to get it out of the lab and make it accessible and useful for most. In that respect, the new Harmful Substances Fellow have received a small media training to help them share their research in 3 minutes, from something somehow technical to a non-scientific audience. Trust me, it's difficult. We hope their 3-minute pitch will grab your attention and enrich your understanding of the issues they deal with, which are varied as glyphosate, textiles, photovoltaic technologies, pollution, etc. You will find on their slides the Twitter at the end of this. If you want, you can contact them and ask them more questions. If you tweet about it, don't forget the hashtag #ShareYourAXAResearch or tag at AXA Research Fund. Enjoy the presentations. To start, the very first presentation of the day is going to be from Ona Freudenthal from Luxembourg Institute of Science and Technology, and she will talk about how to improve regulation of the health impacts of some substances of concern. And Oona. Hi, Oona.
Hello.
How are you?
I'm very good, thank you. A bit nervous, but it's going to be good.
Yeah, thank you for being the very first one and starting the 3-minute speech event. Whenever you're ready, you can start.
Okay, great, thank you. So, have you ever wondered what does a polar bear have in common with a non-sticky frying pan in your kitchen? No one? Well, what if I told you that both contain persistent man-made forever chemicals that would, even if we stopped the use of them tomorrow, still be found in the environment and in our bodies for generations to come? These forever chemicals can also be found in other consumer products than just frying pans. They are found in products that we consume every single day, such as in cosmetics and in clothing. And what if I told you that some of these forever chemicals are toxic for our reproduction system, meaning that they might affect our ability to have babies, and some of them may even cause cancer and other diseases? And these forever chemicals are just one example of harmful substances that surround us. The examples are plenty, and after all, everything we touch is made of chemicals. That's why it's very important to know the risks posed by these substances, as well as to know where they can be found. Fortunately, though, governments and authorities have put in place environmental laws that protect us from some of the most hazardous effects of harmful chemicals. And efforts are constantly being made to create new ways to protect us through regulations. But it's not easy for the legislators to assess which chemicals need most urgent action from the point of view of consumer and environmental protection. Sometimes it's challenging for decision makers to find information on the hazardous properties and the risks and the whereabouts of harmful compounds. And sometimes the data is so plenty and so diverse that it's difficult to make sense of it because the information does not fit well together for a decision to be formed. It's like when you have an Android phone and your partner has an iPhone, and it really bugs you that you can't use the same charger for your phones, although the purpose of both chargers is the same, to get the phone charged. Or when your smaller brother has Duplos and you have the normal big boy Legos, and you can't collaborate to build a joint fortress because the blocks just don't fit together. That's why my passion during this project is to help authorities and insurance companies to match the data available on harmful substances so that we can use this information more efficiently to make decisions and better control the risks of such chemicals on the environment and on the human health. By taking action, we might be able to save the future generations of polar bears from hazardous effects of these forever chemicals, as well as save lives by avoiding cancer and other diseases originating from the use of such substances. Thank you.
Thank you, Una. Thank you very much, and well done. So now we are having a new question. Yeah, next question is, can daily product exposure impact your reproductive health? And it's what Luke Govers from the University of Melbourne, Australia, will talk about for the next 3 minutes. Luke, are you ready?
Yep, going terrific.
Okay, so it's your turn now.
All right, thank you very much. Let me just get the slide up, sorry. Okay, awesome. Fantastic. All right, everyone, I'm going to be talking today about a particular issue that a lot of you have probably never heard of before, and that is a condition called hyperspadias. Now, chances are a lot of you have never heard of it before or never talked about it before, but it's actually the most common abnormality in males and actually affects 1 in 125 males in the world. So a lot of you are probably asking, well, what actually is hyperspadias? Now, hyperspadias basically is an abnormality in the male penis where the urethra opening, the place where urine comes out of, is actually in the wrong position. Now, males with hyperspadias, when they're born, generally require some sort of surgery, and this actually occurs quite early in their life. However, later in life, males with hyperspadias actually have an increased risk of things like testicular or prostate cancer and also have a significantly reduced fertility. So it's actually quite hard for males with hyperspadias to actually conceive children naturally. Now, as I said, this is one of the most common conditions in males and affects 1 in 125 males in the world. But the most alarming aspect of this condition is that in the last 50 years alone, we've actually seen a 50% increase in the number of males being born with hyperspadias. And the large issue around this is actually due to our increased exposure to a wide variety of different chemicals that can harm our fertility and reproduction. So on my slide, I've actually got quite a wide variety of chemicals, and these chemicals can come in a wide variety of sources, from things like plastic bottles or the packaging of food to things like cosmetics or toys that children play with. The most common ones that we see is actually the chemicals sprayed on fruit and vegetables. Now, you might be thinking, okay, well, what does this actually do with anything related to this very common abnormality in males? Well, we actually suspect quite heavily that these chemicals that we're exposed to on a daily basis are actually driving the cause of hyperspadias in the world. But we've actually got a major problem, and that is that there are tens of thousands of chemicals that we are exposed to on a daily basis, and we don't really have the methods to test every single one of them quickly. Now, conventional testing takes 1 to 2 years to actually see whether or not a chemical can potentially cause hyperspadias. But the research that we're conducting at the University of Melbourne and funded by the AXA Research Fund is we're actually creating a whole new technique, a whole new way that we go about testing these chemicals. And ultimately, we can take a 1 to 2 year study to something that's 2 to 3 months. Our primary goal of all of our testing is to try and find what are the chemicals that are causing this major condition in males. And so we start thinking about ways that we can limit them and start to find new alternatives that we can use instead of these hazardous substances. Thank you very much.
Thank you very much, Luke. So our next one is Chang He. Hi, Chang
Hello.
He. So you're from the University of Queensland, Australia, and you're going to share your research on new ways to measure exposure to emerging contaminants. Right?
Yes.
Are you
Thank
ready?
Yes, I'm ready.
Amazing. We're going to put up your slides and you can go.
Thanks. All right. Do you remember when was the last time that you had your health check? And do you remember what test have you done during that check? I had mine a few weeks ago, and here's my result. My doctor checked a few things such as blood pressure, cholesterol, glucose, etc. However, all of them are about biological things, nothing really about chemicals such as organic pollutants. Then you may ask, why there's no such things for chemicals? Well, the quick answer is that we do not know what and how to monitor them. And this is what my research about. Look at my health check result. The first thing I saw is items that have been checked during the health check, which is on the left side. It is the same story for my research. My colleagues and I have created a list of chemicals that are suspected to be linked with human health. On my health check result, apart from the items, I'm actually more concerned and interested in the values of these items. Same for my research. I have developed an analytical method for chemicals in different human tissues. These methods allowed us to provide such values for chemicals in our body. In the end of my health check, my doctor told me that all the items that have been checked are in the acceptable range. That is a super exciting message, isn't it? But the definition of acceptable range did not work so well in my research. A few years ago, some people from Qantas, the biggest airline company in Australia, approached us asking if we can help them to measure the concentration of one chemical in their staff, because someone complained that they inhale in the chemical during their work on the airplane. After some work in the lab, I showed them the result. I said, look, the concentration of this chemical in your human are 3 times higher than in general population. I was super excited about this result, but they stopped me immediately and asked, what does that mean? Can people get sick because of the chemical at this concentration? I said, I'm sorry, I don't know. I was super upset and frustrated. Look, this is a good example of missing the acceptable range of chemical in our body, and this is what I am working on right now. My entire research is to create a health check form for chemical pollutants, including chemical items, results, and acceptable range, just like something we did for the biological things in our health check. Thanks.
Thank you, Chang. Very interesting. Everyone, I'm really happy everyone can manage to do it in 3 minutes, and it's really amazing to see all your research. So we have 3 done already, and we are going to leave you, Chang, to welcome Sejja. Hi, Sejja.
Hey!
So you're from the universities of Turku, Finland, right?
Yes.
And you're going to talk to us about how to mitigate the threat of microplastic in aquatic environments.
Indeed, yes.
Yes. So it's all yours. The slide is going to appear, and then I'm gone, and you have your 3 minutes.
Thank you. Thank you very much. So we know it's a warm Friday on July 22, but did you know that lake muds can keep record of time too? After hundreds of years from now, the lake muds can actually tell to scientists that the summer of 22 was warm. This is because all the changes occurring in the vicinity of the lake leave their fingerprints in the muds. In Northern Europe, like here in Finland, snow and ice covers the landscape during winter, but the summers are still hot and sunny, or at least it's warm. The large differences between the seasons makes precipitation to fall either in form of snow or water. Equally, seasons control which type of material settles down on the lake floor. As a result, we get brown and black pairs of layers in the muds that look much like tree rings. We can take a sample from the lake bottom, count the layers, and know exactly on what year these muds were deposited. So now, at the very moment, microplastics has awakened our concern. Microplastics are plastic fragments and fibers of sizes less than half a centimeter. They originate mostly from decayed plastic consumables that break down in nature due to physical abrasion or, for example, sunlight. The use of plastic materials has increased drastically since last 80 years, and unfortunately, that can be seen in our natural environment at the moment, although the health risks remain still uncovered. Huge plastic dewars are rolling in the world's oceans, and microplastics has been found everywhere, from deepest marine basins to Himalayas, from water that we drink, from soil where we grow our crops, to the air that we breathe. Microplastics are found even in human body, not only in human guts, but also from human blood. To estimate the future health risk, we need to be able to predict how fast the amount of microplastic pollution entering our biosphere is growing. Here, the lake muds step to the picture. We will extract microplastics from the natural annual mud layers to be able to tell exactly how much plastics was deposited on a square meter each individual year and how the accumulation rate has been changed during last 80 years. Knowing how much plastic has been produced every year, we can build scenarios to estimate future pollution rate. We can make predictions for future like we have done to understand climate change. And this project is one step on this path. Thank you.
Thank you, Sejja. Thank you very much. So now we're going to leave you and we're going to go from microplastic to natural fiber. Are they that clean? And this is what Thomas Stanton, from your research fellow in Loughborough University, right? And you're working closely at the University of York as well, with
Correct.
the University of York, to your fellowship. And you're going to talk to us about that, right?
Yes.
Okay, so we put on your slide, yes, and you have 3 minutes now.
Thank you very much. So have you ever bought a natural fibered garment, something made out of cotton or wool, thinking it would be more environmentally friendly than a plastic fiber garment such as polyester or nylon? I'm going to leave you with that and hopefully make you think twice about that in the future. So it shouldn't be news to you that big bits of plastic cause harm. We know that plastic is ingested by animals and that it can entangle them, causing all sorts of harm. And you might have also heard of microplastic pollution, not just from the presentation that came before myself. So a microplastic is anything smaller than 5 millimeters in its largest dimension. And to give you an idea of scale, that's about the size of one of the studs on top of a Lego brick. What you might not have heard about, though, is plastic microfiber pollution. So these are the textile fibers that come off your clothes during day-to-day wear. And also when we wash them, they come out of our washing machine and enter wastewater treatment plants, which is what this little green box is meant to show. And some of these are removed in wastewater treatment, but a lot of them pass through into receiving water, so into rivers and lakes and ultimately into the ocean. Now, what recent research has found is that the majority of these textile fibers that we find in the environment are not actually made from plastics. They're made from things like cotton and wool, particularly cotton. What we don't know, though, is how the risks associated with these natural textile fibers compare to those of the plastic ones that are so much more common in the news and in academic research. And so what my AXA Research Fund Fellowship is going to do is look at how natural textile fibers, things like cotton and wool, move through the environment and how they interact with different chemical pollutants that we know are in aquatic environments, including those things like forever chemicals that were mentioned in our first talk, and then compare these interactions and these mechanisms of movement to the plastic fibres that we are all rightly worried about, to see if these natural textile fibres are something that we should also be concerned with. Thank you very much.
Thank you, Thomas. Thanks. So we talked about microplastic, natural fiber, and now it's time to leave you, Tom, and welcome Meng to talk about the ecotoxicity of emerging photovoltaic technologies. Mainly, you are from Queen Mary University of London, right? Welcome.
Yeah.
And you have 3 minutes to share your research.
Okay. The pollution of the fossil fuels to the environment makes us realize that the development of the clean energy is more and more important to our society. At present, solar cell has become one of the most recognized and reliable clean energy. Solar panel can convert solar energy into electricity that we need in our daily life. For example, on the top of the car, on the roof of the house, or on the wall of the building. About 30 square meters of solar panels can meet the electricity demand of a family for 1 year. Imagine how convenient and eco-friendly it would be if solar panels were installed on the roof of our house. But do you really think that solar cells have no harmful to the environment? In fact, usually what people know is appearance. Solar cells often have to use toxic light, which people don't want to touch it. The most promising second generation photovoltaic material, perovskite, usually also contain a certain amount of toxic light. Once their toxic light containing material leak into the environment, they will cause harm to the people's health and the environment. And they may affect the intellectual development of the children. To solve this problem, my research topic is to use non-material and material structure to reduce the light leakage in the solar cell. We will use their new material and the new structure to control the exchange of light and make them more difficult to leak. Even with a small amount of the light leaking, the new structure could make their toxic light containing material more difficult for the body to absorb. At the same time, we also strive to reduce the use of the light, minimize the harmful to the environment and the people during the preparation and later work of the solar panels. The ideal energy source can not only provide clean energy, but also need to maintain the health of the people. Thank you.
Thank you, Meng. Thank you. So now we're going to leave you and we're going back to microplastic with Joseph Livermore. Hi,
Hello.
Joseph.
Good
So you are from Imperial College London, right?
Yes, that's exactly right.
Yeah, and you're going to talk about how we can limit the effect of microplastic exposure for workers in the recycling chain.
Yep.
So the floor is yours. You have 3 minutes.
Thank you very much. By now, you'll have all seen images depicting how plastic waste is impacting our environment. Whether that be a whale who's unfortunately chowed down on a transparent shopping bag, mistaking it for a jellyfish, or a turtle with a plastic straw wedged up their nose. These images are all signs of a system bowing under the pressure of our consumption. Yet plastic consumption continues to increase, with global production being 367 million tonnes as of last year. These entities, depending on their use span upon consumption, are discarded. And what we do and don't do with them at this point has and can impact our environment and our health. To curtail the release of plastics, more circular economic supply chains have been proposed, increasing the importance of collection and separation of this plastic waste. Which is where, you may argue, but we recycle now, And I agree, recycling has played a role in reducing plastic's environmental presence. However, fully autonomous separation of this plastic waste stream is a thing of the future. And yet, at present, waste separation is underpinned by manual labour. Scientific studies conducted in these locations where the manual labourers sort materials have been found to be incredibly dusty environments. For context, these environments are 4 orders of magnitude greater than air pollution on London's busy streets. But the characterisation of pollutants and their effects on health in these occupational settings has been understudied. Therefore, the aim of my AXA Fellowship is to conduct a pilot study to employ the scientific methods the team and I at Imperial College London have developed, and my interest in health-based sciences to understand how these chemical constituents, microplastic particles and fibres being one of them, contribute to respiratory health impacts observed in individuals working in these occupations. With this occupation expected to increase in societal importance, as it's the first line of defence against the plastic tide, the outcome of this research will provide workers with pivotal information about how working in such environments can impact their health, and with future work exploring ways in which their health can be protected. Thank you very much for listening.
Thank you, Joseph. Thank you. And you were the last one being physically here. So thank you. Well done to everyone that has already done it. We're going to leave you, Joseph, to thank you.
Bye.
And now we have 2 grantees that couldn't be here either because of field work or because of locations where they are, and it's a bit too early in the morning. So we're going to have pre-recorded sessions. The first one we're going to hear from is Loris Évariste from the National Institute for Agricultural Research, which is the INRAE in France. And the question is, what, glyphosate exposure, what is in for your microbiota? So we're going to put on his video and see you in a few minutes.
For the year spent doing complicated researches, I drawn one main and easy conclusion, which is poop is life. Yes, really, and I'm going to explain you why and how important it is. Almost all of us heard as a kid, or you may also tell it to your kids, as I often do with mine, don't touch this, it is dirty, it is full of microbes, it will make you being sick. So when we talk about microbes, we think about sickness, which is not completely exact. You have in your body 100 times more friendly microbes than in the number of stars in the Milky Way, which is huge. Together, these microbes in your body are called microbiota, and they won't make you being sick, but on the contrary, they will help you being in good health. And me, as a scientist, I study these microbes, which can be found in your poop. Talking about being in good health, we also heard when we were younger, eat your vegetables or drink your soup to grow strong, which is true this time, because the microbes in your body love these vegetables. And if you feed them with healthy food, your microbes will promote your good health. If you do not, this may lead to the occurrence of diseases such as obesity or diabetes. However, some agricultural practices are based on the use of crop protection chemicals products that are used to improve the vegetable production. Among them, there is one called glyphosate, which is widely used around the world, and this chemical can be finally found in the food you eat. So, my research project aims to determine the consequences for your microbiota health of eating food containing this chemical and the consequences for your health associated to this microbiota health alteration. So I try to better understand how to take care of your microbes to help your microbes to take care of you. Thank you.
And this is it for Loris. And we're arriving already almost at the end of this event. I hope you enjoyed, and I hope the 3-minute speeches really piqued your curiosity about those research, and really don't hesitate to follow them on Twitter and go and contact them and ask them more questions. The last one for today is a pre-recorded as well. It's Joan Rodriguez Melo. Is from the Instituto de Agrobiotecnología del Litoral from Argentina. And he will talk about can we develop new approach to replace synthetic fungicides currently used in agriculture. So I'm going to play the video for you right now.
According to the WHO, 200,000 people die each year because of the exposure to agrochemicals. However, in developing countries, even now, there are poor pesticide regulation regimen. This results in a constant pesticide exposure for farmers and in the underestimation of how dangerous are pesticides for human health. Also, there is evidence that farmers and people living in adjacent farming areas are more susceptible to long-term health risks associated with the continuous exposure to these chemical substances. This is a horrible landscape now. So, did you know that with the same technology used to create COVID-19 vaccines, we can create vaccines for plants and replace dangerous agrochemicals in crops? This technology could generate sustainable alternatives friendly to health and environment. My name is Johan Rodriguez. I am a PhD in biological sciences, and my project aims to generate a new family of pesticides for organic peanut production and friendly for environment and human health. For several years, the management of diseases in crops has focused on the intensive application of agrochemicals, which allow fast and effective elimination of pathogens that destroy crops. However, we are compromising our health with it. It has been observed that during the last decade, the amount of agricultural products containing pesticide residues has increased enormously. Nowadays, almost every time we eat, we are indirect consumers of pesticides. It's not just food that reaches our tables, it's also agrochemicals. My project aims to design specific fungicides for peanut pathogens, but it can also be used for any other type of plant and other type of pathogens without causing any effect on other organisms. This is a truly novel solution to reduce exposure to harmful substances. Just summarizing, imagine that by finding the weakness of a specific pathogens for a specific plant species, we can design a vaccine for every pathogen. Now imagine that the vaccine that we design is not dangerous for environment or human health. That's what our project wants to reach. Thank you.
This is it. You've heard all the pitches from all the grantees. So 7 that were there and 2 that were pre-recorded. I didn't introduce myself at the beginning. I'm Elodie Chabrol, and I'm a partner of AXA Research Fund, and I was the one that did the training, so I'm pretty proud of them. A huge congratulations to all of them for managing to explain in a very short time with only one slide something that can be pretty technical to a lay audience, and I think they've done a really good job. So congratulations to all of them. I hope you enjoyed it. And as I said before, don't hesitate to follow them on Twitter and to contact them for questions or anything. You have so links in the chat. The event is going to be available on replay, so just take the link, you can share it, everyone can watch it, so don't hesitate to share it around or to watch it on replay if you watch only a part of it. We have another event later today, so same, you can watch it live or watch it on replay after. If you have any question, you can email with the email that is in the chat right now, and that will be in the description. So AXA Research Fund at axa.com. If you want to check all the grantees and what they are doing and all their projects, there is a page, AXA Research Fund, and you can also subscribe to the newsletter of AXA Research Fund to have all the news directly delivered in your email inbox. This is it. So don't hesitate to, contact, register, everything. If you're interested by their work, huge congratulations to them. See you in an hour or two to the next live, or see you on replay. Don't hesitate to share, and have a really good day. Bye.